Welcome to the deep dive. Today, we're plunging into one of the most, just astonishingly complex structures known to us, the human cerebral cortex.
The incredible wrinkly cap sitting right on top of our brain.
Exactly. It's constantly processing, interpreting, directing everything we do. It's, where your thoughts happen, your memories live, even your personality resides. It's pretty mind-boggling when you actually stop and think about it, isn't it?
It absolutely is. And our mission today, well, it's ambitious. We want to not just survey, but to really try and master the cerebral cortex.
Get a deep, expert-level understanding.
Precisely. We're going to peel back its layers, quite literally, to understand its incredibly intricate structure, its precise functional localization where different jobs get done, and what happens when this highest level of the central nervous system faces challenges.
So the goal is for you, the listener, to walk away with that kind of comprehensive grasp that, the elite professionals in this field possess, connecting all the dots.
That's the idea, connecting every concept, every detail, from the microscopic neuron all the way up to the macroscopic functional area.
And to really hammer home why this depth of understanding matters, we're drawing insights from comprehensive neuroanatomy sources, and we'll keep tying it back to a really fascinating real-world case study.
Ah, yes, the case of the 19-year-old woman.
Right. Involved in a severe car accident, suffered head injuries, and after a remarkable recovery, she later started experiencing these sudden jerking movements of her right leg and foot.
Jacksonian epileptic seizures they were diagnosed as.
Exactly. And her initial leg weakness, and then the very specific way her seizures spread, pointed directly, like a map, to specific damaged areas of her cerebral cortex. So this deep dive is really going to equip you with the knowledge to understand precisely why those symptoms popped up, how insights like that guide diagnosis and treatment, and, what it tells us about the brain's incredible wiring.
Okay, let's unpack this then.
So let's start right at the very beginning. When we talk about the cerebral cortex, what exactly is it?
Well, think of the cerebral cortex as the brain's ultimate command center. It forms a complete convoluted covering over the cerebral hemispheres. It's composed of gray matter.
And that's where the neurons mostly are, right?
That's right, billions of them. The estimate is around 10 billion neurons packed into this relatively thin layer.
10 billion. That's just an astonishing number. And it's not smooth, obviously. It's got all those characteristic folds.
Exactly. Its surface area is vastly increased by those convolutions we call the ridges gyri, and they're separated by these grooves or figures known as sulci.
Ah, okay. Gyri and sulci.
And this folding is crucial. It allows that immense number of neurons to fit into a, relatively compact space inside the skull. Its thickness actually varies a bit, from about 1.5 to 4.5 millimeters.
That thin. Wow.
Yep. It's generally thickest over the crest of a gyrus, the top of the ridge, and thinnest down in the depth of a sulcus, the bottom of the groove. And, like other gray matter in the central nervous system, it's this complex mixture of nerve cells, nerve fibers, the neuroglia, which are the support cells, and of course blood vessels.
So what's its overarching role? In the grand scheme of the central nervous system, where does it sit?
That's the key question. It represents the highest level of the central nervous system. That means it's where the most complex processing happens. But, and this is important, it never works in isolation.
Right. It's always talking to other parts.
Always functioning in association with the lower centers in the brain.
Right.
It receives just vast amounts of information from all our senses, our internal states, and then it responds precisely, bringing about the appropriate changes.
And those responses, they aren't just knee-jerk reactions, are they? They're shaped by our whole lives.
That's absolutely right. Those responses are profoundly influenced by, inherited programs, our basic instincts and reflexes, but also, critically, by everything we've learned and stored throughout our individual lives. Our experiences literally shape how the cortex responds.
Which makes sense.
And this ability for detailed functional localization within the cortex, knowing which bit does what, is incredibly powerful for clinicians. It's how they can accurately pinpoint the location of lesions damage in the brain based purely on the patient's clinical symptoms and signs.
Like in that Jacksonian epilepsy case.
Precisely. That specific leg weakness and the seizure pattern immediately told neurologists where the damage likely was, which part of the cortical map was affected.
It really is like having this highly detailed map that lets you pinpoint exactly where a problem originates. But to really understand that map, we need to zoom right in, down to its fundamental building blocks, the cells and fibers making up the cortex. You mentioned there are 5 main types of nerve cells.
That's right. Let's walk through them. First, probably the most iconic are the pyramidal cells.
Okay.
They're named for their triangular or pyramid-shaped cell bodies. They typically range from, say, 10 to 50 micrometers long. But then you have these absolute giants, the Betz cells.
Oh, I've heard of those.
I
They're
know.
big.
Massive. They could be up to 120 micrometers. And you find these exclusively in a very specific place, the motor precentral gyrus of the frontal lobe. They're key players in motor control.
So these are the heavy hitters sending out the big commands for movement. What's their structure like?
Well, they're quite distinctive. Their pointed apices face towards the pile surface that's the outermost membrane covering the brain.
Okay.
And a thick apical dendrite extends upward from that point, branching as it goes. Then, at their base, basal dendrites spread out laterally into the surrounding neuropil.
Neuropil. What's that exactly?
Think of the neuropil as the dense, incredibly intricate wiring of the brain tissue between the cell bodies. It's packed with axons, dendrites, glial cell processes, and countless synaptic connections.
Got it. So the basal dendrites spread out in that dense mesh.
Exactly. And these basal dendrites are covered in numerous dendritic spines, tiny protrusions, which are key sites for receiving synaptic inputs. And crucially, their axons, the output cables, arise from the base of the cell body. These axons either terminate in deeper cortical layers or, more commonly, they enter the white matter to become one of the brain's major communication pathways.
And those pathways are the projection, association, and commissional fibers you mentioned.
Precisely. Those are the three main types. Maybe we should quickly define those.
Yeah, that would be helpful. They seem pretty fundamental.
Okay. Think of the brain as a massive, sprawling city.
Right.
Projection fibers are like the long-distance highways and railway lines. They connect the cortex to distant brain areas, like the brainstem or spinal cord, sending commands out or bringing information in.
Okay, long distance.
Association fibers are more like the internal city roads and local bus routes. They link different parts within the same cerebral hemisphere, allowing one cortical area to talk to another nearby or further away on the same side.
Got it. Same side communication.
And finally, commissural fibers are the big bridges connecting the two sides of the city. Think Golden Gate Bridge, connecting the two hemispheres. They link the left and right cerebral hemispheres, allowing them to coordinate and share information. The biggest one is the corpus callosum.
That's a perfect analogy. Projection long distance, association same side, commissural bridging the two sides. So Betz cells are like a main station for those projection fibers, sending out those long distance motor commands.
Exactly. They are a major source, although not the only source, of those long projection pathways.
Okay, what's the next cell type after pyramidal?
Next, we have stellate cells, sometimes called granule cells. They're quite small, maybe about 8 micrometers in diameter, with a polygonal, star-like shape, hence the name stellate.
Makes sense.
They have multiple branching dendrites, but their axon is relatively short. It usually terminates on a nearby neuron right there within the cortex. So they're primarily involved in local processing, acting as interneurons.
Okay, more for local communication. What about fusiform cells?
Fusiform cells are distinct because their long axis is oriented vertically, perpendicular to the cortical surface. You find them concentrated mainly in the deepest cortical layers.
Okay.
They have dendrites extending from each pole of the cell. The inferior branches stay within the same deep layer, while the superficial ones ascend all the way up toward the pile surface. Their axon also arises from the inferior part, and like pyramidal cells, it enters the white matter as one of those key fiber types: projection, association, or commissural.
Interesting. And the more specialized cells, you mentioned two more.
Right. We have the horizontal cells of Cajal. These are small, also fusiform or spindle-shaped, but uniquely they are oriented horizontally. You find them primarily in the most superficial layer of the cortex, layer 1.
Ah, okay, so they run parallel to the surface.
Exactly. They have a dendrite extending from each end, and their axon also runs parallel to the surface, making contact with the dendrites of pyramidal cells. This allows for lateral spread of information within that top layer.
And the last cell type.
Finally, the cells of Martinati. These are small, multipolar cells, and you find them present throughout all the cortical levels, from deep to superficial. They have short dendrites, but their axon has a really unique trajectory. It's directed upward toward the pile surface, typically ending in a more superficial layer, often the very top one, layer 1.
So it sends information upwards through the layers.
Correct. And this axon also gives off a few short collateral branches on its way up. So each of these 5 cell types plays a really specific role in processing and transmitting information within the cortex and sending it out to other brain regions.
It's just incredible how many different types of nerve cells exist just within this one part of the brain, each with its own very specialized role and structure. Now, we've talked about the cells themselves, but for communication, they obviously need to connect. That's where nerve fibers come in. You mentioned two main types earlier.
Right. Broadly speaking, we have radial fibers and tangential fibers. Radial fibers run at right angles or perpendicular to the cortical surface. Think of them running vertically. This group includes afferent fibers, that's incoming information, whether it's those long-distance projection fibers, local association fibers, or interhemispheric commissural fibers, all of which are entering and terminating within the different layers of the cortex.
So incoming signals.
Incoming signals, yes. And crucially, radial fibers also include the axons leaving the cortex, the outputs from those pyramidal, stellate, and fusiform cells that travel down into the white matter to become those same projection, association, or commissural fibers.
So radial fibers are basically the brain's internal elevators, moving information up and down through the cortical layers, both in and out. What about the tangential fibers then?
The tangential fibers run parallel to the cortical surface, like the horizontal floors of a building or maybe the wiring running along those floors.
Okay, flat along the layers.
Exactly. These are mostly collateral branches and the terminal endings of those incoming afferent fibers spreading information across a particular layer. But they also include the axons of those horizontal cells and some stellate cells, which are designed for local horizontal communication, as well as collateral branches from pyramidal and fusiform cells that also spread sideways.
And I remember you mentioned something really fascinating about these tangential fibers, about how they sometimes form visible bands.
Yes, that's where it gets really interesting for anatomical identification, especially under a microscope. Tangential fibers are most concentrated in certain layers, specifically layers 4 and 5. Here, they form distinct bundles known as the outer and inner bands of Baillarger, respectively.
Bands of Baillarger.
So these bands are particularly well developed in sensory areas of the cortex. Why? Because that's where there's a very high concentration of incoming thalamocortical fibers, those crucial long-distance projection lines bringing sensory data up from the thalamus to the cortex.
So lots of incoming sensory axons make those bands thicker.
Precisely. And in fact, in the visual cortex, the outer band of Baillarger, the one in layer 4, is so thick, so prominent, that you can actually see it with the naked eye on a brain section.
Really? Wow.
Yes, it's called the stria of Gennari. And it's so characteristic that the primary visual cortex is famously known simply as the striate cortex because of this visible stripe.
It's amazing how these microscopic details, these tiny bundles of fibers, can manifest in macroscopic structures visible to the naked eye. That's incredible. And speaking of structure, that leads us perfectly into the layered masterpiece itself, the cortical architecture. You mentioned earlier that the cerebral cortex is typically divided into 6 distinct layers, numbered from superficial to deep. Let's delve into each one because this is where that processing hierarchy, that vertical organization, really starts to become clear.
Absolutely. Understanding these layers is absolutely key to understanding cortical function. So starting at the very top, closest to the surface of the brain, the pia mater, we have the molecular layer, also called the plexiform layer. That's layer 1.
Okay, layer 1, the top layer.
This is the most superficial layer, and it's characterized by a dense, intricate network, a plexus of mostly tangentially oriented nerve fibers. These fibers come from the apical dendrites of pyramidal and fusiform cells, reaching up from deeper layers, the axons of stellate and Martinati cells projecting upwards, and also incoming afferent thalamic and commissural fibers spreading horizontally. You'll also find those occasional horizontal cells of Kajal here.
So it's mostly fibers and connections.
A lot of fibers, yes. And crucially, this layer is a major site for numerous synapses. It's a zone of intense information exchange, particularly horizontal integration.
So layer 1 is like a high-level processing and integration hub, mostly for horizontal communication. What's beneath that? Layer 2?
Beneath that is the external granular layer, layer 2. This layer contains large numbers of relatively small pyramidal cells and, importantly, many stellate cells, which are often called granule cells, hence the name. Their dendrites reach up into the molecular layer, layer 1, to receive inputs, and their axons either enter deeper layers or can project into the white matter. This layer is thought to be very important for associative learning and receiving inputs.
Then we move down to the external pyramidal layer, layer 3.
Correct. As the name strongly suggests, it's primarily composed of pyramidal cells, and these progressively increase in size as you go deeper within the layer. Their apical dendrites extend all the way up to the molecular layer, layer 1, and their axons are really significant output fibers.
How so?
These axons contribute substantially to the white matter as those projection, association, or commisural fibers we discussed. Layer 3 is often considered a major source of association fibers, linking different cortical areas within the same hemisphere, and also commisural fibers connecting to the other hemisphere.
So layer 3 is a big output layer, especially for communication within and between the hemispheres. What's layer 4 like?
Next is the internal granular layer, layer 4. This layer is really distinct. It's characterized by its closely packed stellate cells, those small local processing interneurons, and it has a very high concentration of horizontally arranged fibers. This is where we prominently find that external band of Baillarger we mentioned earlier.
Ah, right, the one that's super thick in the visual cortex.
Exactly.
Yeah.
Its prominence here marks layer 4 as the primary receiving layer for sensory information coming up from the thalamus. If you want to understand where the bulk of sensory input first hits the cortex and gets initially processed, layer 4 is the key target.
Got it. Input layer. And the next layer down, the ganglionic layer or internal pyramidal layer, layer 5.
This is a truly significant layer, especially for motor output. It contains very large and medium-sized pyramidal cells, along with some scattered stellate and Martinati cells. You also find a large number of horizontally arranged fibers here, forming the inner band of Baillarger. But what makes layer 5 particularly famous, especially in certain areas, is that in the motor cortex, specifically the precentral gyrus, the pyramidal cells here include those very large BET cells we met earlier.
Right, the giants.
The giants. And these BET cells, though relatively few in number, maybe only 25,000 to 30,000 in total, are a direct source for about 3% of the corticospinal or pyramidal tract projection fibers. That means they send direct motor commands all the way down the spinal cord to control voluntary movement.
So layer 5 is essentially the primary executive output layer, particularly for initiating skilled movements. And finally, the deepest layer, layer 6.
That's the multiform layer, sometimes called the layer of polymorphic cells, layer 6. Multiform because the cells here have varied shapes. While the majority are fusiform cells, you'll also find many modified pyramidal cells with triangular or ovoid cell bodies, and also quite conspicuous Martinati cells. This layer is extremely rich in nerve fibers that are either entering from or leaving into the underlying white matter.
So it's like the main interface with the white matter highways.
Exactly. It serves as the main gateway between the cortex and the rest of the brain's white matter communication network. It's also known to be involved in sending feedback projections back down to the thalamus, modulating the sensory information that comes up.
So it's like the main entrance and exit ramp for the entire cortical highway system, and it talks back to the sensory relay station too. Now, while these 6 layers form the typical blueprint, you said earlier that not all areas of the cortex strictly adhere to this exact structure. There are variations.
Absolutely. While the majority of the cortex is what we call homotypical, meaning it possesses all 6 layers pretty much as described, there are very important heterotypical cortex areas where the basic 6 layers can't be as easily recognized. These variations strongly indicate specialized functions.
Okay, can you describe those variations? What are the main types of heterotypical cortex?
There are 2 main types to be aware of. First, the granular type. In these areas, layers 2 and 4, the granular layers, packed with stellate or granule cells, are exceptionally well developed and very dense. Conversely, layers 3 and 5, the pyramidal layers, are poorly developed and tend to sort of merge together.
And what does that structure tell us about its function?
These granular areas are characterized by receiving a high volume of incoming sensory information, specifically those thalamocortical fibers bringing data up from the thalamus. So you find this type of granular cortex in key sensory processing hubs, like the postcentral gyrus for touch, the superior temporal gyrus for hearing, and parts of the hippocampal gyrus. And of course, the primary visual cortex, area 17, is the ultimate example of granular cortex.
Okay, so granular cortex means lots of sensory input processing. What's the other type?
The granular type, here's the opposite situation. The granular layers 2 and 4 are practically absent or very thin. Instead, the pyramidal cells in layers 3 and 5 are densely packed and often very large, especially in layer 5.
So fewer input layers, more output layers.
Exactly. This type of cortex is found in areas designed for significant output, especially motor commands. The precentral gyrus, the primary motor cortex, is the classic example of a granular cortex, giving rise to large numbers of efferent, or motor fibers, heading down the spinal cord. Other frontal lobe areas involved in motor planning are also a granular.
And this difference is actually useful clinically.
Absolutely. This specific histological difference, being able to identify granular versus agranular types under a microscope, is a key characteristic used by pathologists, for instance, to identify whether a small piece of cortical tissue is primarily motor or sensory in function. It's a foundational insight for neurodiagnosis, connecting structure directly to function.
That's a powerful clinical distinction just from looking at cell layers. It really highlights how structure dictates function. And thinking about how these layers function together, you mentioned modern neuroscience has refined our understanding with the vertical unit theory. This suggests the cortex isn't just a flat map, but it's organized into these remarkable vertical units or columns of functional activity. How do these columns work?
It's a really fascinating concept, almost like the brain's internal modular design, sometimes called cortical columns. These functional units are typically quite narrow, maybe about 300 to 600 micrometers wide. In sensory cortex, for example, each column seems to be dedicated to processing a single specific sensory feature or location, maybe processing touch, from a particular tiny spot on your skin, or responding to lines of a specific orientation in your visual field, or a specific frequency of sound.
Wow, so very specific jobs for each column.
Very specific. And a functional unit, this column, extends vertically through all 6 layers, from the cortical surface right down to the white matter. Each unit contains the incoming afferent fibers that bring information to it, a network of internuntial neurons, which are basically interneurons connecting other neurons within the column, and efferent fibers that send the process signals out.
So information flows vertically up and down through this column, getting processed along the way. But does it stay completely contained within that single column?
Not entirely, and that's important. An afferent fiber, bringing information in, can synapse directly with an efferent neuron in an output layer, maybe layer 5, to trigger a relatively quick response. Or, more complexly, it can activate vertical chains of those internuntial neurons within the column, allowing for more elaborate processing before an output is generated.
Okay.
And you're right, excitation isn't always strictly confined. It can spread horizontally to adjacent vertical columns via short axon granular cells, mostly in layer 4, allowing for some local interaction and integration between neighboring columns. Plus, remember those horizontal cells of Cajal we discussed earlier, those ones that run parallel to the surface way up in layer 1?
Yeah, the horizontal ones at the top.
They actually allow for the activation of potentially quite distant vertical units from a single incoming afferent fiber. This lateral spread of information, especially in the upper layers, is thought to be crucial for integrating information over larger cortical areas, perhaps allowing the brain to understand complex sensory input by combining data from multiple columns.
So it's not just a straight up and down flow. There's a huge amount of intricate crosstalk and parallel processing happening both within and between these columns. That's a truly sophisticated system.
Okay, so moving from these microscopic building blocks and their layered architecture up to the macroscopic level, let's talk about mapping the brain. Specialized cortical areas. We often hear about specific Brodmann areas, these numbered regions, but it's important to remember that simple divisions, like saying this area is purely motor and this one is purely sensory, can sometimes oversimplify things, right?
Absolutely. The brain is far more integrated and dynamic than those initial, simpler maps sometimes suggested. We now know many sensory areas are actually more extensive and functionally diverse than originally thought, and you can even elicit motor responses from areas traditionally considered purely sensory, highlighting the interconnectedness.
So the boundaries aren't always perfectly sharp. Not always. Areas are often named initially by their anatomical location, like the precentral gyrus, until more precise functional terminology evolves based on research. And the research has exploded. Extensive work using advanced techniques like electrophysiology, recording electrical activity directly, histochemistry, looking at chemical markers, immunocytochemistry, using antibodies to label specific molecules, EEG, PET scans, measuring metabolic activity, and especially functional MRI, tracking blood flow changes related to activity, has vastly increased our knowledge.
Right, fMRI has been huge.
It has. Now, while much of this yields incredibly detailed factual data that isn't yet directly translated into routine clinical practice every day, it's rapidly advancing our fundamental understanding of how the brain works and, importantly, what goes wrong in disease.
Okay, let's start our tour then with the frontal lobe. This is the big one at the front, famously associated with higher level functions and motor control. The precentral area is a key player here. Where exactly is it, and what's its basic function?
The precentral area is located primarily in the precentral gyrus, which is the ridge just anterior to the major central sulcus. It also includes the anterior wall of the central sulcus itself and extends a bit into the posterior parts of the superior, middle, and inferior frontal gyri. and even wraps around the top onto the supramedial border into the paracentral lobule.
So it's a pretty big chunk of cortex.
It is. And histologically, as we mentioned, it's largely a granular type of cortex. It's optimized for output, featuring prominent pyramidal cells, including those giant bed cells in layer 5.
Right.
These bed cells are most concentrated in the superior part of the precentral gyrus and the paracentral lobule areas controlling the leg and trunk. But remember, while there are maybe 25,000 to 30,000 of them, they actually contribute only about 3% of the corticospinal fibers heading down to the spinal cord. Most direct motor commands originate from the millions of smaller pyramidal cells throughout this entire precentral area.
So the BET cells are important maybe for specific types of control, but they're not the only source of motor commands by any means. And this precentral area is then further divided functionally, isn't it, into primary motor and premotor areas?
Yes, that's a key distinction. The posterior region is the primary motor area, also known as M1 or Brodmann Area 4. It specifically occupies the precentral gyrus itself and extends onto the paracentral lobule on the medial surface.
Okay, area 4 is primary motor.
Electrical stimulation of this primary motor area produces very specific, isolated movements on the opposite side of the body, contralateral control. Interestingly, though, it also controls bilateral movements for certain muscles, like the extraocular muscles moving the eyes, muscles of the upper face, tongue, jaw, larynx, and pharynx. Those tend to get signals from both hemispheres.
And this is where the famous homunculus comes in, right? That distorted map of the body stretched across the cortex.
Exactly. When you map out which part of Area 4 controls which body part, you get this distorted representation of the body, often depicted as a little person. The motor homunculus draped over the gyrus, upside down.
Upside down.
Yes, inverted. Swallowing, tongue, jaw, lips, larynx, eyelid, and brow are represented at the very bottom, near the lateral fissure. As you move up the gyrus, you find areas controlling the fingers, especially the thumb, which has a huge area, then the hand, wrist, elbow, shoulder, and trunk. The hip, knee, and ankle are represented in the highest part of the precentral gyrus near the top of the head. And finally, the toes, anal, and bladder sphincters are located over the edge on the medial surface of the hemisphere within the paracentral lobule.
And what's crucial about this map is the size of the cortical area proportional to the size of the muscle it controls?
No, and that's a key insight into motor control. The area of cortex dedicated to controlling a particular movement is proportional to the skill and precision involved in that movement, not the sheer mass of the muscle.
Ah, okay. That makes sense for fingers and lips.
Exactly. Think about the incredible dexterity of your fingers or the fine movements needed for speech. They have disproportionately large cortical representations compared to, say, the large muscles of your back or thigh. The primary function of Area 4 is to carry out individual, discrete movements. You can think of it as the final station for converting a movement design into actual execution commands sent down the spinal cord.
And it needs information to do that, right? Where does it get its instructions from?
It receives essential FREM fibers, incoming information, and instructions from several key areas. The premotor area right next door, the sensory cortex, telling it about body position, the thalamus, the cerebellum, crucial for coordination, and the basal ganglia, involved in selecting and initiating movements. All these inputs converge on the primary motor cortex to shape the final output.
Okay, that brings us neatly to the area just anterior to it. The anterior region known as the premotor area. This includes Brodmann Area 6 and also parts of Areas 8, 44, and 45. How does this differ functionally from the primary motor area?
This area occupies the anterior part of the precentral gyrus, extending forward into the posterior parts of the superior, middle, and inferior frontal gyri. Histologically, it lacks those giant BET cells found in Area 4, and typically it requires a stronger electrical stimulation than the primary motor area to produce the same movement, suggesting it's not as directly connected to the final motor output.
Right.
Like the primary motor area, it also receives crucial inputs from the sensory cortex, the thalamus, and the basal ganglia.
So if the primary motor area executes movements, what's the premotor area's main job?
Its core function is thought to be involved in planning and sequencing movements. It helps to store programs of motor activity based on past experience. Think learned skills like playing an instrument or typing, and then it helps program the primary motor area, selecting the appropriate sequence of actions. You can think of it as the brain's choreographer or movement planner.
That makes perfect sense, like setting up the sequence before hitting go on the primary motor cortex.
Exactly. It's also particularly involved in controlling coarser postural movements, getting your body ready for a movement, partly via its extensive connections with the basal ganglia.
Okay, that clarifies the primary motor versus premotor distinction. Beyond that, What other specialized motor-related areas are in the frontal lobe?
Well, there's the supplementary motor area, SMA. This is situated on the medial surface of the frontal gyrus, just anterior to the paracentral lobule, where the leg is represented in M1. Stimulation here can also elicit movements of contralateral limbs, but usually requires an even stronger stimulus than the premotor area.
And is it essential?
Interestingly, removal of the SMA typically causes no permanent loss of movement, although there might be temporary difficulties with initiating movements or performing complex sequences. This suggests its role is more involved in the internal planning, preparation, and initiation of movements, perhaps especially for sequences learned from memory rather than direct execution.
We also have areas specifically for eye movement in the frontal lobe, don't we? The frontal eye field.
Yes, absolutely. The frontal eye field, SEF, includes parts of Brodmann areas 6, 8, and 9. It's typically located extending forward from the facial area of the precentral gyrus into the middle frontal gyrus. Stimulation here causes conjugate movements of the eyes, meaning both eyes move together smoothly, especially voluntary movements toward the opposite side.
So this controls where we choose to look.
Precisely. The FEF controls voluntary scanning movements of the eye, for example, when you're consciously deciding to look across a room or scan a page of text. This is independent of visual stimuli just catching your attention. It is, however, connected to the occipital visual area, which handles the more automatic, involuntary following of moving objects. So there's a fascinating interplay between voluntary control from the front and stimulus-driven control from the back.
And then famously, one of the most critical areas for human communication, Broca's area, essential for speech production.
Absolutely vital. The motor speech area of Broca encompasses Brodmann areas 44 and 45. It's located in the inferior frontal gyrus, specifically tucked between the anterior and ascending rami and the ascending and posterior rami of the lateral fissure, or Sylvian fissure.
And it's usually on one side.
Critically, this area is profoundly dominant in the left hemisphere for the vast majority of individuals, both right and left-handers. Damage specifically to Broca's area in the dominant hemisphere leads to what's called Broca's aphasia, or expressive aphasia, historically called aphemia. This is the loss of the ability to produce fluent speech.
So they can understand language fine, but can't speak properly.
Exactly. Patients can still think clearly. They can often understand spoken and written language quite well, and they may even be able to write, but they struggle immensely to articulate words. Speech becomes slow, effortful, ungrammatical, often consisting of short phrases or single words. It's as if the motor programs for generating speech are damaged. Broca's area connects directly to adjacent primary motor areas to stimulate the coordinated action of muscles in the larynx, mouth, tongue, soft palate, and respiratory system, all necessary for the incredibly complex act of forming words.
So it's the orchestrator of the physical act of speaking. And then there's the largest and perhaps most enigmatic part of the frontal lobe, the prefrontal cortex. This is where our higher functions like personality and decision-making really come from.
Precisely. The prefrontal cortex, BFC, is everything anterior to the precentral motor and premotor areas. It comprises Brodmann areas 9, 10, 11, and 12, and it's an incredibly extensive area, including most of the superior, middle, and inferior frontal gyri, the orbital gyri on the underside, the medial frontal gyrus, and the anterior part of the cingulate gyrus.
Wow, it's huge.
It really is. And it's characterized by its incredibly rich afferent and efferent pathways. It connects to virtually all other cortical areas, as well as extensively with the thalamus, especially the dorsomedial nucleus, the hypothalamus, the corpus striatum, part of the basal ganglia, and the cerebellum, via connections through the pons, the frontopontine fibers, and of course commissural fibers, like the forceps minor traveling in the genule of the corpus callosum, ensure these prefrontal areas are densely interconnected between the two hemispheres.
So it's massively connected. What does all that connectivity allow it to do?
This vast connectivity is why the PFC is so deeply concerned with the highest levels of cognitive function and behavior. It's critical for an individual's personality, their depth of feeling and emotional regulation, initiative and motivation, planning and foresight, working memory, holding information online, abstract reasoning, and judgment and decision-making. It's where we integrate complex information, anticipate consequences, control impulses, and essentially exercise executive control over our thoughts and actions.
It's truly the seat of many of our higher-level human characteristics. Okay, let's shift our focus now back towards the central sulcus, but this time behind it, to the parietal lobe. This lobe is primarily associated with processing sensory information from the body, right?
That's its main role, yes. And the key region here, just posterior to the central sulcus, is the primary somesthetic area, also called the primary somatic sensory cortex, or S1. This corresponds to Brodmann areas 3, 1, and 2.
Okay, areas 3, 1, 2 is S1.
It occupies the postcentral gyrus, the ridge immediately behind the central sulcus, and also extends over the top onto the posterior part of the paracentral lobule on the medial surface of the brain. Histologically, particularly the anterior part, area 3, right up against the central sulcus, is a classic granular type of cortex.
Meaning it's optimized for receiving sensory input.
Exactly. It's packed with small, granule stellate cells in layers 2 and 4, and it boasts that broad and obvious outer band of Bayarger in layer 4, indicating massive thalamic input. The posterior parts, areas 1 and 2, have fewer granular cells and are more involved in further processing of that sensory information. This whole S1 area receives direct projection fibers, carrying sensory information touch, pressure, vibration, pain, temperature, proprioception from the ventral posterior lateral, VPL, and ventral posterior medial, DPM nuclei of the thalamus. Those are the critical relay stations for all bodily sensory information heading to the cortex.
And similar to the motor cortex on the other side of the central sulcus, is there a sensory body map here as well, a sensory homunculus?
There absolutely is, and like the motor map, it's also largely inverted. The opposite half of the body is represented contralaterally here. The pharyngeal region, tongue, and jaws are mapped at the very inferior end of the postcentral gyrus, near the lateral fissure. Then, as you move superiorly up the gyrus, you find a face, fingers, especially the thumb and index finger, again with huge areas, hand, arm, trunk, and thigh. And just like the motor map, the leg and foot are represented over the top, on the medial surface in the posterior paracentral lobule. The anal and genital regions are also represented down there on the medial side.
So another homunculus, but this one for sensation. And again, is the cortical area apportioned by the size of the body part or by its sensitivity?
Great question. Just like with the motor cortex, the amount of cortical real estate devoted to a body part in S1 is related to its functional importance and the density of sensory receptors it possesses, rather than its sheer physical size.
So big areas for lips and fingertips again.
Exactly. You have disproportionately large areas dedicated to the face, lips, thumb, and index finger, because these regions have an incredibly high density of sensory receptors and are crucial for fine discriminative touch and exploration. While most sensations are processed contralaterally from the opposite side of the body, it's worth noting that some sensation from the oral region actually goes to the same side cortex as well, and sensory information from the pharynx, larynx, and perineum projects bilaterally to both hemispheres.
Interesting. What happens functionally when that sensory information arrives at S1, primarily in layer 4?
When those afferent fibers carrying sensory signals arrive, they primarily excite neurons in layer 4, the main granular input layer. From there, the signals spread vertically through the cortical column for further processing in other layers. An absolutely fascinating aspect is that axons originating from neurons in layer 6, the deepest layer of S1, actually project back down to provide feedback to the lower sensory relay stations, particularly the thalamic nuclei, VPL, VPM, but also even further down to the brainstem and spinal cord.
So the cortex talks back to the relay stations. What does that feedback do?
This feedback is largely inhibitory. It plays a crucial role in modulating the flow and intensity of the sensory input reaching the cortex. It helps the brain to filter out irrelevant background noise, sharpen contrast between stimuli, and focus attention on important sensory signals. It's a key mechanism for refining sensory perception. Also, it's important to note that the most anterior part of the postcentral gyrus, area 3A, located right within the central sulcus, receives abundant afferent fibers, specifically from specialized receptors in our muscles, muscle spindles, tendons, Golgi tendon organs, and joints are proprioceptors.
So information about body position and movement.
Exactly. And this crucial proprioceptive sensory information directly influences the primary motor cortex, area 4, just across the sulcus, providing essential feedback for ongoing motor control and adjustments.
That's a lot of sophisticated refinement happening right there at the first stage of cortical sensory processing. Is there a secondary sensory area as well, like there's a secondary motor area?
Yes, there is a secondary somesthetic area, S2. It's located along the superior lip of the posterior limb of the lateral fissure, sort of tucked underneath S1. It's much smaller and generally considered less functionally critical, or at least less well understood, than S1.
Okay.
The face area is represented anteriorly within S2 and the leg area posteriorly. Interestingly, the body seems to be bilaterally represented here, though the contralateral side remains dominant. Neurons in S2 respond particularly well to transient cutaneous stimuli like light brushstrokes or tapping, suggesting perhaps a role in detecting changes in touch or contributing to tactile learning and memory, but its precise function is still debated.
And then beyond S1 and S2, there's the synesthetic association area, this sounds like, where we really start to make sense of all this raw sensory data, right? Putting it all together.
That's exactly right. The synesthetic association area, corresponding roughly to Brodmann areas 5 and 7, is located in the superior parietal lobule, just posterior to S1, and it extends onto the medial surface of the hemisphere as well. It has extensive connections with S1 and other sensory areas throughout the brain.
And its main function?
Its main function is to receive and integrate different sensory modalities. It takes the raw data about touch, pressure, temperature, pain, and proprioception from S1 and combines it, relates it to past experiences, and interprets it. This integration is what enables crucial abilities like stereognosis.
Stereognosis, what's that again?
That's that incredible ability you have to recognize common objects placed in your hand without looking at them. You can identify a key, a coin, or a cotton ball just by feeling its shape, size, texture, temperature, and weight. That requires integrating all those different sensory inputs, which happens here.
Ah, right. It makes sense of the combined feelings.
Exactly. It constantly relates current sensory information to past sensory experiences stored in memory, allowing for interpretation and recognition. It also plays an absolutely vital role in assembling a conscious appreciation of the body image, or body schema. That's your internal sense of where each part of your body is in space, how your limbs are positioned, and how your body relates to its environment. This body image is absolutely crucial for planning and executing coordinated movements. And consistently with other sensory maps, the right side of the body's sensory world and image is primarily represented in the left parietal association cortex, and vice versa.
So it's not just feeling a sensation, but understanding what that sensation means in the context of an object or your own body. Okay, moving from touch and body sense to the very back of the brain, we arrive at the occipital lobe, our dedicated visual processing powerhouse. The primary visual area, V1, or Brodmann area 17, is the main entry point for everything we see, isn't it?
It is indeed. The primary visual area is situated mainly in the walls of the posterior part of the calcarine sulcus, which is a deep fissure on the medial surface of the occipital lobe. It sometimes extends slightly around onto the occipital pole, the very back tip, or the lateral surface of the brain.
And didn't you say this one is visibly different?
Yes. Macroscopically, on a stained brain slice, Area 17 is often easily recognized by the relative thinness of the cortex overall, and, most distinctly, by that prominent white strike running through its middle layers, the visual sotria, or sotria of Gennari.
Right, the striate cortex.
Exactly. And microscopically, confirming its role as a primary sensory receiving area, it's a classic granular type of cortex with densely packed stellate cells in layer 4 and relatively few large pyramidal cells, especially in the output layers. It's built to receive and analyze incoming visual data.
And how does it receive that visual information from our eyes? What's the pathway?
It receives its main afferent fibers directly from the lateral geniculate nucleus, LGN, which is the primary visual relay center in the thalamus. These fibers, forming the optic radiation, have a rather fascinating looping path. They first pass forward into the white matter of the temporal lobe. Meyer's loop carries information from the lower visual field, and then they sweep back to terminate in the occipital lobe in area 17.
Okay. And how does the input from the two eyes get organized?
It's very precisely organized to represent the visual world. Each primary visual cortex receives fibers originating from the temporal half of the ipsilateral retina, that's the outer half of the retina on the same side, and the nasal half of the contralateral retina, the inner half of the retina on the opposite side.
Wow, okay, that's specific. Temporal, same side, nasal, opposite side. What does that mean for what we actually see?
This complex crossover pattern ensures that the entire right half of the visual field, from both eyes, is represented in the visual cortex of the left cerebral hemisphere, and the entire left half of the visual field is represented in the visual cortex of the right cerebral hemisphere.
So one side of the brain sees the opposite half of the world, and how precisely is that visual field mapped within Area 17 along the calcarine sulcus?
It's remarkably precise, a concept called retinotopy. The superior retinal quadrants, which actually see the inferior field of vision because the eye's lens inverts the image, project to the cortex in the superior wall of the calcarine sulcus. Conversely, the inferior retinal quadrants, which see the superior field of vision, project to the cortex in the inferior wall of the calcarine sulcus.
Okay, so upper visual field to lower bank, lower visual field to upper bank. What about central versus peripheral vision?
That's also mapped very specifically. The macula lutea, the small central part of our retina responsible for our most acute, high-detail vision, has a disproportionately huge representation in the cortex. It's mapped primarily in the most posterior part of Area 17, near the occipital pole, and accounts for perhaps as much as 1/3 of the entire primary visual cortex area. The peripheral retina, providing less sharp vision, terminates in progressively more anterior concentric circles along the calcarine sulcus. This detailed mapping is why specific patterns of visual field loss can pinpoint the exact location of a lesion within the visual pathway or cortex.
That's incredible precision. And the emphasis on the macula makes sense given how important central vision is. What surrounds this primary visual area? Is there a secondary area for vision too?
Yes, surrounding the primary visual area, V1, is the secondary visual area, which includes Brodmann areas 18 and 19. These areas are sometimes collectively referred to as extraindustriate visual cortex, and they are further subdivided into many functionally distinct areas, like V2, V3, V4, V5MT, etc., involved in processing specific aspects of the visual scene. Broadly speaking, this secondary visual cortex receives major afferent fibers from Area 17, as well as input from other cortical areas and the thalamus, particularly the pulvinar.
And its overall function. If V1 detects the basic features, what does V2 and beyond do?
Their critical function is to further process the visual information received from V1, extracting more complex features like shape, color, movement, and spatial relationships, and crucially to relate this visual information to past visual experiences stored in memory. This is what enables us to recognize and appreciate what one is seeing. It's the difference between V1 detecting lines and colors, and the secondary areas allowing you to recognize that pattern as a familiar face, a specific object, or a written word.
So V1 sees, V2 and beyond recognize and interpret. Is there an eye field back here too, similar to the frontal eye field we discussed earlier?
Yes, an occipital eye field is thought to exist, likely within the secondary visual area, perhaps Area 19. Stimulation here tends to cause involuntary reflexive conjugate deviation of the eyes, especially towards the opposite side. This field seems particularly associated with automatically tracking or following moving objects in the visual field.
Ah, so this is different from the frontal eye field.
Yes, it's important to contrast it with the frontal eye field, FEF. The FEF controls voluntary intentional scanning movements of the eyes, independent of direct visual stimuli. The occipital eye field mediates reflexive, stimulus-driven movements, like your eyes automatically jumping to follow a bird flying past. It's likely connected to the superior colliculus in the brainstem, which is involved in visual reflexes. So one field for voluntary looking, the other for automatic following.
That distinction makes sense. Okay, now let's move forward in the brain from the occiput down below the lateral fissure to the temporal lobe. This lobe is crucial for hearing, memory formation, and aspects of language, right?
Absolutely. A major player here is the primary auditory area, A1, corresponding to Brodmann areas 41 and 42. This is where sound information first arrives in the cortex.
And where is it located?
It's situated mostly on the superior surface of the temporal lobe, deep within the lateral sulcus, Sylvian fissure, specifically on the transverse temporal gyri, often called Heschl's gyrus, or gyri as there can be more than one. Histologically, Area 41, the core primary area, is a granular type of cortex, again optimized for receiving incoming sensory data, while the surrounding Area 42 is more homotypical and considered mainly auditory association cortex.
Okay, and where does it get its input from?
The projection fibers delivering auditory information to this area primarily rise from the medial geniculate nucleus, MGN, which is the main auditory relay center in the thalamus. These fibers form the auditory radiation, which travels up through a part of the internal capsule to reach Heschl's gyrus.
And is sound frequency mapped here, similar to how visual space is mapped in V1? Is there a tonotopic map?
Yes, there is. The primary auditory cortex exhibits tonotopy. The anterior part of Heschl's gyrus primarily receives and processes low-frequency sounds, while the posterior part receives and processes high-frequency sounds. It's like a piano keyboard laid out along the gyrus.
That's sweet. What happens clinically if this area is damaged on one side? Does the person go deaf in one ear?
That's a crucial clinical point. Because the auditory pathways are partially crossed, but also have significant ipsilateral connections, a unilateral lesion, meaning damage to the primary auditory area on only one side, produces only a partial hearing loss, hypoacusis, in both ears. However, the hearing loss is generally greater in the contralateral ear, the ear opposite the lesion.
So you don't get complete deafness in one ear from a cortical lesion?
Not typically. The main functional deficit often reported from a unilateral lesion isn't profound deafness, but rather difficulty in locating the source of a sound in space, which requires comparing inputs from both ears accurately. Bilateral destruction of both primary auditory areas, however, does cause essentially complete cortical deafness.
Okay, so A1 is about discerning the basic features of sound, like pitch and volume, and maybe localization. What about making sense of those sounds, recognizing a melody or understanding speech?
That's where the secondary auditory area, also known as the auditory association cortex, comes in. This corresponds mainly to Brodmann area 22, located just posterior to the primary auditory area on the superior temporal gyrus, extending back along the lateral sulcus. It receives impulses from the primary auditory area, A1, and also directly from auditory nuclei in the thalamus.
And its role is interpretation.
Exactly. This area is thought to be necessary for the interpretation of sounds, not just hearing them, but understanding what they mean. Is that a car horn, a piece of music, or someone speaking? It's also crucial for the association of auditory input with other sensory information and with memory.
And critically, this secondary auditory area, particularly in one hemisphere, is strongly linked to one of the most vital language centers, Wernicke's area.
Yes, absolutely fundamental for language. The sensory speech area of Wernicke is typically localized in the left dominant hemisphere for most individuals. It's located primarily in the posterior part of the superior temporal gyrus, overlapping with area 22, but it also extends posteriorly around the very end of the lateral sulcus into the adjacent parietal region, specifically the angular and supramarginal gyri.
Okay. And it's connected to Broca's area, right?
Yes, it's intimately connected to Broca's motor speech area in the frontal lobe via a large bundle of association fibers called the arcuate fasciculus. This allows for communication between language comprehension and production centers. Wernicke's area itself receives highly processed input fibers from the visual cortex in the occipital lobe via the angular gyrus, important for reading, and from the auditory cortex, area 22, in the superior temporal gyrus, important for understanding spoken words.
So it's a major convergence zone for sensory information relevant to language.
Exactly. This confluence of highly processed sensory information is why Wernicke's area permits the understanding of both written and spoken language. It enables a person to hear words or see written text and comprehend their meaning. It's the brain's primary language comprehension center. Lesions here lead to Wernicke's aphasia, receptive aphasia, where patients can produce fluent but often nonsensical speech and have profound difficulty understanding language. This area is profoundly important as the convergence point for somatic, visual, and auditory association areas, integrating all those sensory streams into meaningful language comprehension.
It really is the brain's language comprehension hub, where all that sensory language input finally makes sense. Okay, before we move on to how things go wrong with lesions across the cortex, are there any other specific cortical areas we should quickly highlight, like taste or balance?
Yes, a few important ones to mention. There's the taste area, gustatory cortex, believed to be located partly in Brodmann area 43. This is situated at the very lower end of the postcentral gyrus, extending onto the superior wall of the lateral sulcus and into the adjoining anterior part of the insula.
The insula again, okay. And the pathway for taste?
It involves ascending fibers originating from taste receptors on the tongue traveling up through brainstem nuclei, like the nucleus solitarius, to the ventral postremedial nucleus, VPM, of the thalamus, and then finally projecting from the thalamus to this cortical taste area.
So that's where we actually register and perceive the sensation of taste. What about balance and our sense of spatial orientation? Where does that get processed?
That's handled by the vestibular area. Its exact location is still debated somewhat, but it's believed to be situated near the region of the postcentral gyrus, concerned with face sensation, and possibly extending into an area just opposite the auditory area in the superior temporal gyrus, near the posterior end of the lateral fissure.
And its function?
It's deeply concerned with the conscious appreciation of head position and movements in space, derived from input from the vestibular system in the inner ear. It also plays a vital role in influencing eye movements, like the vestibulo-ocular reflex that stabilizes vision when your head moves, and coordinating trunk and limb muscle activity, all crucial for maintaining posture and balance.
And finally, let's revisit the mysterious insula. We mentioned it for taste and possibly speech articulation. It's tucked away, almost hidden, isn't it?
It is. The insula, or insular cortex, is an entire lobe of cortex, buried deep within the lateral sulcus, Sylvian fissure, forming its floor. You have to pull the temporal lobe down and the frontal parietal lobes up to see it. The posterior part tends to be more granular in structure, receiving sensory inputs like taste, visceral sensation, while the anterior part is more granular, suggesting output or integrative functions.
And its connections and overall role.
Its fiber connections are complex and still being fully mapped and understood, but it connects widely with limbic structures, autonomic centers, sensory areas, and motor planning areas. Beyond the roles in taste and possibly coordinating speech articulation that we mentioned, it's increasingly thought to be important for interoception, sensing the internal state of the body, processing emotions, especially negative ones like disgust, contributing to consciousness and self-awareness, and integrating sensory, emotional, and cognitive information. It's a really fascinating and complex area that's getting a lot more attention now.
So definitely not just a blank slate, despite being hidden away. Now, we've walked through these incredibly specialized primary sensory and motor regions, but as you pointed out, they only form a relatively small part of the entire cortical surface area. What about the rest of the brain, the large majority that isn't directly primary motor or sensory?
That's what we broadly term the association cortex. As you said, the primary sensory, granular, and primary motor angular areas together form only a fraction, maybe 20–25% of the total cortical surface. The remaining vast territories are primarily homotypical in structure, meaning they possess all 6 cellular layers in a more generalized, less extremely specialized form than the primary areas.
Okay. And historically, these were just called association areas.
Yes, and for a long time, their precise functions were, frankly, poorly understood. The simple idea was that they just served to associate or integrate information coming from the primary sensory areas to prepare for output from the primary motor areas. But that view is now seen as far too simplistic.
How has that understanding evolved? What do we think these large association areas are really doing?
We now understand that they have multiple complex inputs and outputs, connecting not just with primary areas, but also extensively with each other, with the thalamus, especially higher order nuclei like the pulvinar and dorsomedial nucleus, and with limbic structures involved in emotion and memory. They are greatly concerned with much more sophisticated functions like complex perception, attention, language processing, spatial awareness, executive functions, planning, decision-making, memory retrieval, and integrating information across different modalities to form coherent representations of the world and guide behavior. They're not just simple relay stations. They are hubs for higher-order processing.
So they're doing the really complex integration and thinking. Can we identify specific large association areas?
We generally recognize 3 main, very large regions of association cortex.
Okay. You already highlighted the prefrontal cortex earlier, which we know is crucial for executive functions, personality, judgment, and initiative. What about the other 2 major ones?
Then there's the posterior parietal cortex. This corresponds roughly to the superior and inferior parietal lobules, including areas 5, 7, 39, and 40. This area is crucial because it integrates visual information, flowing forward from the occipital cortex with somatosensory input, touch, pressure, proprioception, coming from S1 in the anterior parietal lobe, and also auditory information.
And what does this integration achieve?
This multimodal integration is vital for creating a spatial map of the world around us and locating objects within it relative to our own body. It's essential for guiding movements in space, like reaching for an object, for spatial attention, directing focus, and for forming those concepts of size, form, and texture necessary for stereognosis. As we discussed, it also assembles that conscious appreciation of the body image. Understanding where our body parts are damaged here can lead to neglect syndromes. And again, the right side of space in the body is primarily processed in the left parietal cortex, and vice versa.
That explains why damage here can lead to such profound deficits in spatial awareness and even recognizing one's own body parts. What's the 3rd major association area?
That would be the temporal association cortex, located in the inferior and middle temporal gyri, extending toward the temporal pole. This area is heavily involved in processing high-level visual information related to object recognition, the what pathway, as opposed to the parietal where pathway, and face recognition. The anterior part of the temporal lobe, near the hippocampus and amygdala, is also thought to play a significant role in the storage and retrieval of previous sensory experiences and declarative memories for facts and events.
So like a sensory uploading bank and object recognition center?
In a way, yes. Electrical stimulation in parts of the temporal lobe, particularly during surgery for epilepsy, has sometimes been reported to cause patients to experience vivid recall of past events, seen objects, or heard music, highlighting its role in accessing long-term memories and linking sensory information to stored knowledge.
That's absolutely fascinating. So these association areas are where the brain does its most complex integration and interpretation. This brings us to another fascinating high-level concept, cerebral dominance, or hemispheric specialization. We have two cerebral hemispheres that look almost identical anatomically, but functionally, they're not completely interchangeable, are they?
That's one of the brain's most intriguing organizational principles.
Yeah.
Anatomically, as you say, the two cerebral hemispheres are remarkably similar, almost mirror images. And the major nervous pathways project largely contralaterally to the opposite side and fairly equally to identical cortical areas on both sides. Furthermore, the brain's incredible communication network, particularly the massive cerebral commissures like the corpus callosum, connecting most cortical areas, and the smaller anterior commissures connecting parts of the temporal lobes and olfactory structures, allows for a constant massive flow of interhemispheric information transfer. The two sides are always talking to each other.
So they look the same and they talk to each other constantly, but despite that, some activities are clearly dominated by one side or the other.
Exactly. Despite the anatomical similarity and the continuous crosstalk, certain complex cognitive functions are predominantly processed or controlled by one hemisphere more than the other. This is called lateralization, or cerebral dominance. The classic example, of course, is language.
Right. So the left hemisphere is typically the dominant one for language.
For the vast majority of people, yes.
Yeah.
The left hemisphere is typically dominant for processing the components of language, like phonology, syntax, semantics, and for producing speech, Broca's area, and understanding language, Wernicke's area. It's also strongly associated with analytical thought, logical reasoning, mathematical calculation, and, interestingly, handedness. Most right-handers are left hemisphere dominant for language.
Okay. And what about the right hemisphere, the so-called non-dominant one? What does it specialize in?
The right hemisphere, while typically non-dominant for language components, excels in other crucial domains. It's generally considered dominant for spatial perception, things like navigating, understanding maps, visualizing objects in 3D, and constructing things. It's also critical for recognizing faces, processing emotional expression, both perceiving it in others and producing it ourselves, understanding prosody, the emotional tone and rhythm of speech, and processing music and non-linguistic sounds. It's more involved in holistic, synthetic, and intuitive processing.
So they really do have complementary specializations. What's the prevalence of this typical left dominance pattern?
The prevalence is quite striking. As mentioned, more than 90% of adults are right-handed, and they are almost universally left hemisphere dominant for language. Even among left-handers, the majority, perhaps 70%, are still left hemisphere dominant for language, while the rest show either right hemisphere dominance or more bilateral representation. Overall, for speech and language functions specifically, about 96% of the adult population is left hemisphere dominant.
That's very consistent. Is there any known anatomical basis for this? You mentioned handedness earlier.
It's subtle, but there are some reported anatomical asymmetries. For example, an area in the temporal lobe involved in language processing, the planum temporal, is often physically larger on the left side than the right, even in infants. Some research also suggested, as you noted, that perhaps slightly more descending motofibers cross the midline from the left hemisphere in the pyramids of the medulla, potentially contributing to the prevalence of right-hand dominance. But these anatomical correlations are still debated and likely don't tell the whole story.
So the million-dollar question, is this dominance something we're strictly born with, programmed from the start, or does it develop over time?
That's a crucial point, especially from a clinical perspective regarding brain injury and recovery. The current understanding is that while there might be some innate predispositions or biases, newborn hemispheres are believed to have considerable equipotential capabilities. That means they start with roughly similar potential to develop various functions, including language.
So it's not fully fixed at birth?
No. Hemispheric dominance, particularly for complex functions like language, appears to develop gradually and solidify throughout childhood, becoming largely fixed only after the first decade of life, perhaps around puberty.
And that developmental window is really significant clinically.
Immensely significant. It means that if a young child, say before the age of 5 or 6, suffers significant damage to their typically dominant left hemisphere, the plasticity of the developing brain often allows the intact, opposite right hemisphere to take over language functions remarkably well. The child can often learn to speak and comprehend language almost normally, perhaps becoming left-handed if the motor cortex was also affected. This kind of large-scale functional transference and recovery is almost impossible for an adult whose brain organization and dominance patterns are already firmly established. The brain truly has a remarkable but time-limited capacity for adaptation and plasticity, especially when it's young and still wiring itself up.
That's a really powerful example of the brain's resilience and adaptability. And that adaptability, or sometimes the lack of it in adults, becomes especially clear when things go wrong. Which brings us naturally to when things go wrong. Clinical insights from lesions. What have we learned about cortical function from studying what happens when specific areas are damaged?
Studying the effects of human brain lesions, whether they result from strokes, vascular accidents, tumors, surgical interventions like removing epileptic foci, or traumatic head injuries, has been absolutely fundamental to understanding brain function. It's often tragic for the individual, but it provides invaluable insights into which parts of the brain are necessary for which abilities.
Right, it helps map function by seeing what's lost.
Exactly. These studies also reveal the remarkable, though often incomplete, ability of the human cerebral cortex to reorganize itself functionally and allow for some degree of recovery even after significant lesions, especially if the damage occurs slowly or early in life. We know the cortex is the final receiving station for sensory information, deeply involved in discrimination, interpretation, and relating current input to past memories. And the intricate interplay between the cortex and deeper structures, like the basal nuclei and thalamus, is also absolutely vital for normal function, and disruptions here also cause problems.
Okay, let's walk through some specific examples based on the areas we've discussed. Let's start with motor cortex lesions. What happens if there's damage to the primary motor area, area 4?
Lesions specifically confined to the primary motor cortex, area 4, result primarily in paralysis, or at least weakness, paresis, of the muscles on the contralateral side of the body. The deficits are typically most pronounced for fine, skilled movements, particularly involving the distal extremities, the fingers, hand, and foot. Gross movements might be less affected. Generally, the paralysis from an area 4 lesion is considered more severe and debilitating than that caused by a lesion restricted to the secondary motor areas alone.
So if the primary area is about execution, what happens if only the secondary motor area, like the premotor cortex, area 6, is damaged?
Lesions of the secondary motor area, area 6, by itself, if area 4 is spared, tend to produce more difficulty in performing skilled movements and complex sequences, a condition sometimes called apraxia, but often with relatively little loss of raw muscle strength. The patient might be clumsy or unable to coordinate movements smoothly.
And what if both are damaged, which is probably more common in something like a large stroke?
Yes, exactly. Combined primary and secondary motor area lesions, which often occur together due to shared blood supply, typically lead to the most complete and severe contralateral paralysis, affecting both the planning programming, area 6, and the execution, area 4 of movements.
And this directly relates back to our opening case study, Jacksonian epileptic seizures. Can you remind us how that specific type of seizure illustrates motor cortex function?
Absolutely. Jacksonian epileptic seizures are a classic example of what happens with an irritative lesion, meaning a lesion like a scar or small tumor that isn't destroying tissue but is instead overstimulating the neurons, specifically affecting the primary motor area, area 4. The hallmark is the seizure march.
The more
Yes. The convulsive activity, the jerking, begins in the specific body part whose representation lies within the irritated cortical area, like the foot in our case study, if the irritation is in the paracentral lobule. Then, as the abnormal electrical irritation physically spreads across the primary motor area map, the seizure spreads sequentially to involve adjacent body regions, following the order of the motor homunculus. So it might march from the foot, up the leg, to the trunk, perhaps to the arm and hand. This predictable spread is a direct reflection of the somatotopic organization of area 4.
That's a really clear, almost dramatic illustration of that cortical map in action. Now, what about muscle spasticity? We often hear about spasticity in cases of stroke or brain injury involving motor areas. How does that arise?
Muscle spasticity, that state of increased muscle tone, hyperactive stretch reflexes, and velocity-dependent resistance to movement is a complex phenomenon. Interestingly, a very discrete lesion limited only to the primary motor cortex, area 4, often results in relatively little change in muscle tone, or possibly even flaccid paralysis initially.
Really? Not spasticity?
Not necessarily from a pure area 4 lesion. However, larger lesions, which are much more common clinically, and especially those involving both the primary and secondary motor areas, areas 4 and 6, do typically result in muscle spasm or spasticity.
Why the difference? Why does involving area 6 lead to spasticity?
The generally accepted explanation involves the balance between different descending motor pathways. The primary motor cortex, area 4, gives rise to the direct corticospinal pyramidal tract, which tends to facilitate muscle tone, especially in anti-gravity muscles. However, the secondary motor cortex, area 6, gives rise to other descending pathways, often considered part of the extrapyramidal system, though that term is a bit dated, which project indirectly via the brainstem and tend to have an overall inhibitory influence on muscle tone, helping to modulate and smooth movements. So when you have a lesion that destroys area 6 along with area 4, you lose that crucial inhibitory influence from area 6 pathways, leaving the tone-enhancing effects of other pathways, including perhaps some from area 4 itself and brainstem pathways, unopposed. This imbalance leads to the characteristic state of spasticity.
That's a great example of how damage isn't just about losing a function, but also about disrupting the balance between excitation and inhibition. Now, what about lesions affecting the frontal eye field, FEF? How do they impact eye movements?
Lesions of the FEF have predictable effects. With destructive lesions, like a stroke damaging the FEF, the tonic drive from the intact FEF on the other side becomes unopposed. This causes the eyes to deviate towards the side of the lesion, and the patient will have difficulty voluntarily looking towards the opposite side.
So they look towards the damaged side.
Yes. Interestingly, involuntary tracking movements, like following a moving object, often remain unaffected because, as we discussed, that function involves the occipital visual cortex and pathways through the brainstem, bypassing the voluntary control of the FEF. Conversely, irritative lesions like seizure activity starting in the FEF cause overstimulation. This drives the eyes forcefully toward the opposite side of the lesion during the seizure.
Okay, that makes sense. Let's move to speech area lesions, which can be particularly devastating for communication. We talked about Broca's and Wernicke's areas. What happens specifically if Broca's area in the dominant hemisphere is damaged?
Damage specifically to the Broca motor speech area, typically in the left inferior frontal gyrus, areas 44-45, results in Broca's aphasia, or expressive aphasia, euphemia. As we noted, this is primarily a loss of the ability to produce fluent grammatical speech. Patients retain the ability to comprehend language reasonably well, though subtle comprehension deficits can exist, and they can think clearly, but they struggle immensely to articulate words and form sentences. Speech is often slow, effortful, telegraphic, missing small function words, and poorly articulated. They're often aware of their difficulty and frustrated by it.
And what about the Wernicke sensory speech area? That's primarily about understanding language, right?
Exactly. Lesions affecting the Wernicke sensory speech area in the dominant hemisphere, posterior superior temporal gyrus, area 22, often extending into parietal areas, produce Wernicke's aphasia or receptive aphasia. This is characterized by a profound loss of the ability to understand both spoken and written language. The patient can typically hear sounds and see words perfectly well, but the linguistic meaning is lost.
Can they still speak?
Interestingly, yes. Because Broca's motor speech area is usually unaffected, their speech production remains fluent, often rapid and effortless. However, the content of their speech is often nonsensical. They may use incorrect words, paraphasias, made-up words, neologisms, or string words together in a grammatically confused way, sometimes referred to as word salad. And remarkably, patients with Wernicke's aphasia are often completely unaware of their own mistakes and their inability to comprehend a condition called anosognosia, which can make communication incredibly difficult and frustrating for everyone involved.
That's a profound difference. One struggles to speak but understands. The other speaks fluently but understands little and doesn't realize it. What if a large lesion damages both areas?
When you have a large lesion, often from a major stroke affecting the middle cerebral artery territory, that damages both Broca's and Wernicke's areas and the connections between them, that results in global aphasia, a devastating near-total loss of both speech production and language comprehension.
Are there other, more specific language deficits from lesions in nearby areas?
Yes. We mentioned lesions involving the insula can sometimes lead to apraxia of speech, a difficulty in planning and sequencing the articulatory movements needed for speech, often producing sounds close to the target word but not exactly correct, and damage specifically to the dominant angular gyrus, area 39, and the parietal lobe, which acts as a crucial relay between visual association areas and Wernicke's area, can selectively disrupt the pathway for reading. This can result in alexia, the inability to read, often accompanied by agraphia, the inability to write, even if spoken language comprehension and production are relatively spared.
So very specific language deficits can result from lesions in very specific cortical locations or pathways. Now, we touched on the prefrontal cortex, PFC, as the seat of personality and executive function. What happens clinically when that highly integrated area is damaged?
Damage to the prefrontal cortex, especially the large dorsolateral and orbitofrontal regions, typically does not produce a marked loss of general intelligence as measured by standard IQ tests. Motor and sensory functions are also usually spared. However, the impact on higher-level cognitive and emotional functions and on personality itself can be profound and debilitating.
How so? What changes?
The PFC is absolutely crucial for abstract thinking, planning, foresight, judgment, working memory, inhibiting inappropriate responses, and regulating social behavior and emotional expression. Tumors or traumatic destruction, like in the famous case of Phineas Gage, or more modern examples like the professor you mentioned who lost social inhibition, can result in a severe loss of initiative, motivation, abulia, and judgment. Patients may become apathetic, unable to plan for the future, or make sound decisions.
And emotional changes too.
Yes, the emotional changes are often very striking. Depending on the location of the damage, patients might show a tendency towards inappropriate euphoria or irritability, a marked lack of empathy or concern for consequences, impaired social awareness leading to nonconformity with social norms, impulsivity, and sometimes a surprising carelessness about personal dress and appearance. Their underlying personality structure seems fundamentally altered.
That's such a stark illustration of how this brain area defines so much of who we are as individuals. And you mentioned a potential connection to schizophrenia earlier.
Yes. The prefrontal cortex receives a very rich dopaminergic innervation from the ventral tegmental area in the midbrain. Dysfunction in this dopamine system, particularly hypofunction, too little activity, in the dorsolateral PFC, has been hypothesized for decades to contribute significantly to the negative symptoms, like apathy, lack of motivation, flattened affect, and cognitive deficits, like problems with working memory and executive function, seen in schizophrenia. Functional imaging studies, like PT scans, have often shown reduced blood flow or metabolic activity in the prefrontal cortex of schizophrenic patients, especially when they are attempting tasks that heavily rely on executive functions. It's likely part of a more complex circuit dysfunction, but the PFC seems central.
That's a really important connection. And historically, weren't there surgical procedures targeting the prefrontal cortex?
Yes, tragically in many ways, but reflecting the desperation to treat severe mental illness and intractable pain before modern drugs were available. Procedures like frontal leucotomy or lobotomy, developed in the 1930s and 40s, involved surgically cutting the white matter fiber tracts connecting the prefrontal cortex to other brain regions, particularly the thalamus and limbic system. Later, frontal lobectomy, the actual removal of parts of the frontal lobe, was sometimes performed.
Those sound incredibly drastic. What was the intended goal?
The aim was essentially to reduce the patient's emotional responsiveness and suffering by disrupting the prefrontal cortex's ability to process emotional information, anticipate the future, or engage in complex introspection. The idea was to disconnect the thinking part from the feeling part. For example, a patient with terminal cancer suffering from intractable pain might still report feeling the physical sensation of pain after a leucotomy, but they would allegedly no longer worry about it or experience the associated emotional anguish and fear of the future. It was also used, often controversially and with devastating side effects on personality and initiative, for severe psychiatric conditions like untreatable depression, anxiety, or obsessive-compulsive disorders.
It sounds like it came at a very high cost.
An extremely high cost in terms of personality, creativity, and overall function for many patients. Thankfully, it's crucial to emphasize that these procedures are now almost entirely obsolete, largely following the development of modern psychotropic medications like antipsychotics and antidepressants, and more targeted neurosurgical approaches like deep brain stimulation for specific disorders that offer far more effective and less destructive treatment options.
That's definitely a relief. Okay, let's shift back to sensory cortex lesions. If the primary synesthetic area, S1, is damaged, what happens to our sense of touch, pressure, pain, and temperature?
Lesions involving S1, areas 3, 1, 2, result in contralateral sensory disturbances, meaning sensory loss or impairment on the opposite side of the body. These deficits are often most severe distally in the hands and feet, reflecting the larger cortical representation of these areas.
What kind of sensory loss? Is everything gone?
Not necessarily completely. While the ability to finely discriminate sensations is usually lost, some degree of crude awareness of painful tactile touch and thermal temperature stimuli may eventually return after a period. This recovery of crude sensation is largely attributed to the preserved function of the thalamus, which can mediate basic awareness of these modalities even without the cortex. However, the patient typically cannot accurately judge degrees of warmth or cold, cannot precisely localize where a tactile stimulus is applied on their body, cannot recognize objects by touch, asterignosis, and cannot accurately judge the weights of objects held in the hand. Fine discriminative aspects of sensation are lost. A loss of muscle tone, hypotonia on the affected side, can also sometimes be an associated symptom, highlighting again the intimate link between sensory feedback and motor control.
So basic sensation might remain thanks to the thalamus, but the cortical interpretation and discrimination are gone. What about lesions to the secondary sensory areas, like S2?
Interestingly, isolated lesions confined just to the secondary cellular. Some static area, S2, typically do not cause easily recognizable or consistently reported sensory defects that clinicians can readily identify at the bedside. Its precise role in conscious perception remains somewhat unclear.
But lesions to the sympathetic association area in the superior parietal lobule areas 5 and 7, that has major consequences, right?
Absolutely. Lesions here profoundly interfere with the ability to integrate and interpret complex sensory information, particularly from touch and proprioception. This leads directly to asterignosis, which we defined earlier, the inability to appreciate or recognize the texture, size, and form of objects simply by handling them with the eyes closed. The patient might feel the object's individual features, but cannot synthesize them into a recognizable whole. They might be unable to identify a key, a coin, or even distinguish between different textures like cotton versus sandpaper purely by touch.
And you also mentioned earlier that this area is crucial for our body image. What happens if that's disrupted?
Yes. Destruction involving the posterior parietal cortex, particularly in the non-dominant, usually right hemisphere, can severely interfere with the conscious appreciation of the body image or body schema, specifically for the opposite, usually left side of the body in space. This can lead to a range of fascinating and bizarre phenomena collectively known as neglect syndrome.
Neglect syndrome.
Yes. The patient essentially fails to recognize or pay attention to the left side of their own body and the left side of the world around them. They might fail to wash, dress, or shave the left side of their body, might bump into objects on their left, ignore food on the left side of their plate, or even deny that their own left arm or leg belongs to them, a condition called asomatic It's as if half of their world and half of their own body has simply ceased to exist in their conscious awareness. It's a really stark reminder of how the brain actively constructs our sense of self and our relationship to the space around us.
That's truly disorienting and almost hard to imagine. Okay, moving back to the visual area lesions in the occipital lobe. What happens if the primary visual area, V1, area 17, is damaged?
Lesions destroying the primary visual area, particularly those affecting the cortex around the posterior calcarine sulcus, result in a very specific pattern of blindness called contralateral homonymous hemianopia.
Okay, break that down. Contralateral homonymous hemianopia.
Contralateral means affecting the visual field opposite to the side of the brain lesion. Homonymous means the same part of the visual field is lost in both eyes. And hemianopia means loss of half of the visual field. So a lesion in the left visual cortex causes blindness in the entire right visual field of both eyes, and vice versa.
So you lose one whole side of your visual world. Is it always the entire half?
Often, yes, if the entire V1 is affected. However, sometimes the very central part of the visual field corresponding to the macula might appear to be relatively spared, a phenomenon called macular sparing. The exact reason for this is debated. It might be due to slight compensatory eye movements during testing, or possibly because the macular representation at the occipital pole has a somewhat dual or overlapping blood supply, making it more resilient to certain types of vascular damage.
Can you get smaller visual field losses?
Absolutely. Lesions affecting only the upper half of area 17, the upper bank of the calcarine sulcus, processing the inferior visual field, cause an inferior quadrantic hemianopia blindness in the lower quarter of the contralateral visual field. Conversely, lesions of the lower half of area 17, lower bank, processing superior visual field, cause a superior quadrantic hemianopia, loss of vision in the upper quarter. And specific lesions affecting just the occipital pole, where the macula has its large representation, can produce a central scotoma, a blind spot right in the very center of vision, severely impacting reading and detailed sight.
So the pattern of visual field loss is extremely informative about the lesion location. What are common causes?
Common causes of such lesions include vascular disorders like strokes, especially involving the posterior cerebral artery, tumors growing in the occipital lobe, or traumatic injuries like penetrating head wounds. Gunshot wounds were historically a major source of information about visual cortex localization.
Okay, what about lesions affecting the secondary visual area, areas 18, 19, and beyond? If V1 is intact, can the person still see?
Yes, if V1 is intact, the person can still detect visual stimuli. They aren't blind in the primary sense. However, lesions in the secondary or association visual areas result in visual agnosia, a loss of the ability to recognize or interpret what they're seeing, particularly in the opposite visual field if the lesion is unilateral.
So they can see an object but don't know what it is?
Exactly. They might be able to describe its features. It's long, metallic, has teeth at one end, but they fail to recognize it as a key. This is because the connection between the visual percept and the stored knowledge or past visual experiences about that object has been disrupted. There are different types of visual agnosia, like prosopagnosia, inability to recognize familiar faces, often associated with bilateral lesions in the temporal association cortex.
Fascinating. Now let's consider auditory area lesions in the temporal lobe. What happens if the primary auditory area A1 is damaged?
As we touched on earlier, because of the bilateral nature of auditory pathways, unilateral lesions confined to the primary auditory area, A1, cause only slight bilateral hearing loss, though the loss is generally greater in the ear opposite the lesion. The main functional deficit, however, is often a significant difficulty in localizing the source of a sound in space, as this requires comparing the timing and intensity differences between the two ears, a process likely impaired by the cortical damage. Bilateral destruction of both primary auditory areas, of course, results in essentially complete cortical deafness.
And how about lesions to the secondary auditory area, area 22?
Lesions here, particularly in the dominant hemisphere, result in an inability to interpret the meaning of sounds, even though they can be heard perfectly well. This can manifest as auditory agnosia, inability to recognize non-speech sounds like a doorbell or a dog barking, or more specifically as word deafness, also known as pure auditory verbal agnosia.
Word deafness.
Yes. In this condition, the patient can hear spoken words clearly but cannot understand their meaning, as if listening to a foreign language they don't know. However, they can often still read and write normally, and their own speech production might be unaffected. It's a disruption specifically in decoding the auditory signal into meaningful linguistic units, likely due to damage in Wernicke's area or its connections from A1.
It's amazing how specific these deficits can be. And just to tie it all back one last time to that crucial concept of brain development and plasticity, cerebral dominance and damage in a clinical context. You emphasized how the age at which dominance becomes fixed is so important for recovery, especially for language.
It really is a cornerstone of pediatric neurology and rehabilitation. The age at which cerebral dominance becomes firmly established, generally considered to be around the first decade, is profoundly important for predicting prognosis after brain injury. As we said, significant damage to the dominant, usually left hemisphere, before a child has fully developed and lateralized language, often allows a remarkable plasticity of the developing brain to kick in. Speech and language functions can frequently develop and be maintained, albeit perhaps with some subtle differences, in the remaining intact, usually right, hemisphere.
But that doesn't happen in adults.
That kind of large-scale functional transference and near-complete recovery is much, much more difficult, often impossible, in older individuals once hemispheric specialization and dominance patterns are firmly established. The adult brain retains some plasticity, certainly, which is the basis for rehabilitation, but its capacity for wholesale reorganization is significantly reduced compared to the developing brain. It really highlights why understanding the principles of brain development and plasticity is so critical for both basic neuroscience and clinical practice.
The brain's capacity for adaptation, especially in childhood, is truly profound. Okay, shifting gears slightly for our final section, let's explore the underlying mechanisms that allow all this cortical function. Consciousness, sleep, and epilepsy. The electrical activity of the brain. We know neurons communicate electrically. How do we actually measure or observe that collective activity in the cortex?
That's right. The cortex is buzzing with electrical activity. Individual neurons, as we know, maintain a negative resting potential across their membrane, typically around -60 or -70 millivolts relative to the outside. When they're sufficiently stimulated, they generate brief electrical impulses called action potentials, during which the membrane potential rapidly reverses and overshoots zero. Even at rest, this membrane potential continuously fluctuates due to the constant but variable barrage of incoming synaptic inputs from other neurons.
So it's never truly silent. How do we record this collective activity?
We can record the summed electrical activity of large populations of cortical neurons. Electrocorticograms, ECOG, are highly precise recordings made by placing electrodes directly on the cortical surface, usually during brain surgery for epilepsy or tumor removal. This gives a very clear picture of local activity. More commonly and non-invasively, we use electroencephalograms, or EEGs.
Right, the skull cap with all the wires.
Exactly. EEGs are made by placing an array of electrodes on the scalp. These electrodes record the incredibly tiny electrical potential changes, typically around 50 microvolts, so millions of a volt, that manage to filter through the skull from the underlying cortical activity. It's like listening to the faint hum of a city from a distance.
So we're literally listening to the brain's synchronized electrical rhythms.
Right.
And are there characteristic patterns or frequencies in these EEG rhythms?
Yes, absolutely. In normal, healthy individuals, several characteristic frequency bands are recognized in the EEG, and their prominence changes depending on the person's state of arousal or activity. The main ones are alpha rhythms, around 813 hertz, typically seen when someone is awake but relaxed with their eyes closed. Beta rhythms, faster, 13 hertz, usually associated with active thinking, alertness, concentration, or anxiety. Theta rhythms, 4 to 7 hertz, often seen during drowsiness or light sleep, but also during certain cognitive tasks. And delta rhythms, slowest, 4 hertz, which are prominent during deep, slow-wave sleep, but abnormal if dominant during wakefulness in adults.
And these patterns are clinically useful.
Immensely valuable. EEG abnormalities like abnormal slowing, sharp waves, or spikes are clinically crucial for diagnosing and monitoring a range of cerebral conditions, including identifying the focus of seizures and epilepsy, detecting brain tumors or abscesses, which can disrupt normal rhythms, assessing brain damage after injury or stroke, and evaluating sleep disorders. In fact, an electrically silent EEG, a flat line indicating no detectable cortical electrical activity, is a critical component in the clinical determination of cerebral death or brain death.
That's a profound diagnostic tool, reading the brain's own electrical language. Building on this idea of brain activity, what exactly is consciousness? It seems like such a fundamental yet incredibly elusive concept to define neurobiologically.
It is arguably one of the biggest challenges in neuroscience and philosophy. From a clinical perspective, though, we can define a conscious person as someone who is both awake and aware of themselves and their surroundings. And achieving this state requires the active integrated functioning of at least two main parts of the brain.
Okay, what are those two essential components?
First, you need the reticular formation, particularly the ascending reticular activating system, ARS, which is a network of neurons located throughout the brainstem, medulla, pons, midbrain. This system projects widely up to the thalamus and cortex and is essential for maintaining wakefulness, simply being physiologically awake and alert rather than asleep or comatose.
So the brainstem provides the power switch for wakefulness.
You could think of it that way, yes. But wakefulness alone isn't consciousness. The second crucial component is the cerebral cortex itself, particularly the association areas we discussed, interacting extensively with the thalamus. The cortex is necessary for awareness, that is, the content of consciousness, perceiving stimuli, having thoughts and feelings, understanding language, forming intentions, and meaningfully interacting with the environment.
So wakefulness from the brainstem enables awareness by the cortex.
Exactly. You need both working together. For example, eye-opening is primarily a brainstem-mediated function reflecting wakefulness, but producing meaningful speech is a complex cerebral cortex function reflecting awareness. And here's an interesting clinical point. Drugs that produce general anesthesia and unconsciousness are thought to act primarily by selectively depressing the activity of the reticular activating mechanism in the brainstem and thalamus, effectively turning off wakefulness and thereby preventing awareness. Conversely, stimulant drugs that increase wakefulness often act by enhancing the activity of the system.
So wakefulness and awareness are distinct components, and consciousness requires both. How do clinicians describe the progressive loss of consciousness? Are there defined stages?
Yes, clinicians often observe a continuum of declining consciousness, which frequently reverses in the opposite order upon recovery. It typically starts with lethargy, where the patient is drowsy but can be aroused easily. Their speech may be slow, voluntary movements are diminished and slow, and eye movements might be sluggish.
Okay, lethargy first, then.
This can progress to stupor. In stupor, the patient is much harder to arouse. They might only respond briefly, perhaps with groans or simple movements, if stimulated strongly, often with painful stimuli. They generally don't speak spontaneously, voluntary movements are nearly absent, their eyes are usually closed, and there's very little spontaneous eye movement.
Deeper than stupor.
Deeper still is deep stupor, where the patient responds minimally, if at all, even to severe pain, perhaps only with reflex posturing or mass, undifferentiated movements. They will not speak, and there's even less spontaneous eye movement. And finally, this leads to coma.
Coma being complete unconsciousness.
Yes. In coma, the patient is unarousable. They will not speak, and they respond only reflexly to painful stimuli, like withdrawing a limb, or, in deep coma, not at all. Their eyes remain closed, and there are typically no spontaneous movements. These progressively altered states of consciousness usually involve dysfunction of the thalamocortical system and/or the reticular formation, either directly due to bilateral damage or dysfunction, or indirectly due to factors like swelling, herniation, or metabolic disturbances causing widespread depression of these vital areas.
That progression makes sense, but that brings us to a specific state that's often misunderstood by the public. The persistent vegetative state, PVS. How does that fit into this wakefulness awareness spectrum? It sounds like coma, but isn't quite the same.
It's a critical distinction and often a source of confusion and distress for families. In a persistent vegetative state, the patient has suffered severe, widespread damage primarily to the cerebral cortex, rendering it essentially non-functional for awareness. However, their brainstem, including the reticular activating system, remains relatively intact and functional. So what does that mean clinically? They're awake, but not aware.
Exactly. Because the brainstem is working, the patient is awake, their eyes are open, they have spontaneous eye movements, though not tracking. They may move their limbs or head spontaneously, but non-purposefully. They grimace or cry out sometimes. And importantly, they have preserved sleep-wake cycles. They look awake. But because the cortex is devastated, they have no awareness whatsoever of themselves or their environment. They cannot perceive stimuli meaningfully, cannot follow commands, cannot communicate, cannot show any purposeful behavior or sign of conscious thought.
Wow. That must be incredibly difficult for families to see someone who looks awake but isn't there.
It is extremely difficult. This state often follows severe head injuries or profound anoxic cerebral insult, lack of oxygen to the brain, for example, after cardiac arrest. Lay observers may easily mistake the preserved wakefulness, the open eyes, the movements for signs of consciousness, leading to false hope or confusion about the prognosis, which is typically very poor for recovery of awareness in PVS. The key neurobiological concept here is that it's entirely possible to be physiologically wakeful without having cortical awareness. But having awareness is impossible without wakefulness because the cortex simply requires that activating input from the brainstem's reticular formation to generate conscious experience.
That's a really crucial clinical and ethical differentiation. And then there's sleep, which you described as a changed state of consciousness, but definitely not total unconsciousness like coma or PVS.
Precisely. Sleep is a natural reversible state characterized by reduced responsiveness to external stimuli and decreased motor activity. Physiologically, during sleep, parameters change. Pulse rate, respiratory rate, and blood pressure typically fall compared to wakefulness. The eyes often deviate upward, pupils constrict but still react to light. Most tendon reflexes are diminished or lost, and the plantar reflex, Babinski sign, may become extensor, especially in deep sleep.
But you can be woken up.
Absolutely. That's the defining feature. A sleeping person is not unconscious in the sense of coma. They can be awakened relatively quickly by salient stimuli like hearing their name called or a parent hearing their child's cry, even through significant background noise. Sleep is thought to be actively generated by specific brain circuits and facilitated by factors like reduced sensory input, darkness, quiet, and physiological fatigue, which lead to decreased activity of the ascending reticular formation and the thalamocortical activating mechanism, allowing the brain to enter its restorative sleep state, cycling through different stages like REM and slow-wave sleep.
Okay. And finally, let's discuss a condition that is fundamentally characterized by disturbances in the brain's electrical activity, often originating in the cortex. Epilepsy.
Yes, epilepsy is a neurological disorder defined by a predisposition to generate recurrent, unprovoked seizures. A seizure itself is a sudden, transitory disturbance of normal brain physiology, usually involving abnormal, excessive, and hypersynchronous electrical discharges from populations of neurons, typically in the cerebral cortex. This abnormal activity disrupts normal brain function temporarily, ceases spontaneously, and in epilepsy tends to recur.
And seizures aren't all the same, right? You mentioned partial versus generalized earlier.
Correct. Seizures are broadly classified based on where the abnormal electrical activity begins and how widely it spreads. In partial seizures, now often called focal seizures, the abnormal discharge originates within a localized network of neurons confined to one part of one's cerebral hemisphere. Critically, during many types of focal seizures, specifically simple partial seizures, now termed focal aware seizures, the patient typically does not lose consciousness, although their awareness, sensations, movements, or emotions might be altered depending on the location of the seizure focus, like the Jacksonian motor seizure starting at one limb. Other focal seizures, complex, partial, now termed focal impaired awareness seizures, do involve altered consciousness.
Okay, so focal seizures start in one spot. What about generalized?
In generalized seizures, the abnormal electrical activity appears to begin in or rapidly involves large areas of the brain bilaterally, affecting both hemispheres more or less simultaneously from the outset. Because of this widespread disruption, the individual typically loses consciousness abruptly.
Are all generalized seizures convulsive?
No. Some generalized seizures are non-convulsive, such as the classic petit mal or absent seizure, most common in childhood, where the patient certainly stops activity, stares blankly for a few seconds, and is unresponsive, often with subtle eyelid fluttering, before abruptly returning to normal, usually unaware the seizure occurred. However, the most dramatic and widely recognized type is the generalized tonic-clonic seizure, formerly grand bowel.
What happens in that type?
This typically involves a sudden loss of consciousness, often with a cry as air is forced out, followed by a tonic phase where all muscles stiffen rigidly, often causing the person to fall, then a clonic phase characterized by rhythmic jerking or convulsive movements of the limbs. There's often transient apnea, cessation of breathing, sometimes biting the tongue, and frequently loss of bowel or bladder control. The whole seizure usually lasts from seconds up to a few minutes, followed by a period of confusion, drowsiness, and muscle soreness known as the postictal state.
That sounds very intense. And what causes epilepsy? Is it always clear?
For a large proportion of patients with epilepsy, perhaps the majority, the underlying cause remains unknown. This is termed idiopathic or genetic generalized epilepsy, suggesting an underlying genetic predisposition to neuronal hyperexcitability. However, in a significant number of cases, especially those with focal seizures, a specific underlying cause or local lesion can be identified. This might be a structural abnormality like a cerebral tumor, a scar tissue focus resulting from previous head trauma, as in our opening patient case with Jacksonian seizures after her accident, a stroke, a brain infection, or a congenital brain malformation. Identifying such lesions is crucial, as they might require specific treatment beyond just anti-seizure medications.
Wow. We have truly covered an incredible amount of ground today, navigating the astonishingly intricate landscape of the cerebral cortex. We've gone from its most basic building blocks, the neurons and fibers, through its complex layered architecture, mapped out its specialized functional areas across all the lobes, and importantly, delved into the profound clinical implications of lesions, seeing how precise damage leads to very precise, predictable symptoms.
We really have. And hopefully you, the listener, now have a much more comprehensive, integrated framework for understanding how this master control center orchestrates virtually every aspect of our thoughts, movements, perceptions, and even our personality. We've seen repeatedly how this precise, in-depth understanding of these structures and their functions isn't just abstract academic knowledge. It's profoundly vital for making sense of real-world neurological conditions, for guiding accurate diagnosis, and for developing effective treatments, as highlighted by understanding the mechanisms behind our patient's Jacksonian epilepsy and how neurosurgery could potentially target that specific focal point. This deep dive has aimed to equip you with that kind of integrated knowledge that elite professionals use daily.
Absolutely. It connects the microscopic details to the macroscopic functions in the human experience of neurological health and disease. And as we wrap up this deep dive, maybe a final thought to leave you with, consider the brain's truly remarkable capacity for reorganization and recovery that we touched upon several times, the plasticity seen especially in the developing brain after injury, but also present to some degree throughout life. What is this innate ability of the brain, particularly the cortex, to adapt, compensate, and rewire itself? Tell us about the very nature of consciousness, learning, identity, and adaptation itself.
It's a profound question. The cerebral cortex, in all its complexity, clearly isn't just a static, hardwired map laid out at birth. It's a dynamic, ever-evolving frontier that actively shapes itself through experience and possesses an incredible ability to heal, compensate, and learn. What new insights will future research reveal about its profound ability not just to process information, but to fundamentally change its own structure and function in response to the world, and even in response to damage, in ways we are still only beginning to fully comprehend? It remains one of the most exciting frontiers in science.